Thank you very much for the introduction. Okay. My talk will be more about the, let's say, biosensing using graphene and 2D materials. And, it's more like an overview, workshop overview style presentation. And once again, I mean, it's a really huge topic, and it's a little bit so. Whatever I'm presenting here, it's really more like a sprinkle on what is actually happening in the field right now. So, let's start with sort of making a biosensor. What does make a biosensor, and then we'll go to why 2D materials are actually an excellent biosensor. So biosensor, in a general description, would go as something that you can measure in electrical signals, say something you can depict in your computer that you can see physically, and something that you want to measure and analyze, which could be DNA, which could be chemical, which could be your blood composition, glucose, iron, viruses, COVID-19 viruses, could be electricity or cellular activity. What does really make a biosensor are these 2 points in between, the receptors and the transducers. So transducers is where the 2D materials would be coming mainly, and the receptors are chemical recognition elements that would come on top of the biosensors to make the transducer, to make our biosensor specific sensitive to our analyte of choice. And here is exactly why 2D materials are perhaps one of the most amazing materials or elements that can make a biosensor. Say the 4 major elements, and why 2D materials make excellent electrical and electrochemical biosensors are their high electrical mobility, their flexibility and stretchability, so you can also make wearable, as Professor Kimanda just mentioned. But really, well, they're also biocompatible, they're stable, that you can tune the band gaps, so you have like hundreds of those 2D materials. There's very different band gaps, so you can really make very interesting and different specific optical 2D materials, for example. I will not focus on optical too much, mainly on electrical and biochemical this overview. So the last 2 really important parameters on why 2D materials make excellent biosensors are the controlled surface reactive sites and high surface to volume ratio. Why is it important? A biosensor, so let's say you have a electrically conductive graphene or MOS2, and you put it in a liquid to make a biosensor, but these devices would be sensitive to literally anything with the charge. You get a charged molecule, you receive a reaction. This is not a biosensor per se, because it's not specific to a molecule. And to make specificity, you need to functionalize the 2D materials with recognition elements, as I introduced earlier, the receptors. And so let's say I would split these biorecognition elements from the chemistry of the 2D materials to how do you get the recognition element onto in let's say 3 different ways. One, let's say you have a pristine covalent link. So you have atomic surface, which could be carbon, which could be molybdenum, which could be silicon. In the case of silicon, it could be boron, whatever 2D material you're using. They would have their own specific intrinsic bonds. And in ideal case, which is rather impossible, in ideal case, this intrinsic bonds would react to the recognition elements right away, and you have your biosensor in a single step. It is rarely a reality, and you need to functionalize the bond. So between your intrinsic bond, you need to put a linker, which would have a recognition element. So it would react with your intrinsic bond. And on the other side, it would have a functional bond, and this functional bond would then react with the recognition element. This is more common inducing covalent link in your 2D biosensors. A third, and it's a really common approach specifically with graphene, you can make a non-covalent link. So you can have your functional bond, and in this case, the most common molecule is an aromatic pyrene molecule that interacts with graphene non-covalently via pi-pi interaction. but the interaction is pretty strong, so it actually stays on top of it. And you can bind this recognition element on top of graphene to really make a biosensor. And Professor and Corby have introduced a lot of the whole library of the 2D materials. I would just like to zoom in into all of those. And if you look into all of the chemical elements that are there, carbon, boron, silicon, germanium, phosphorine, they would react with all different chemical elements. So we're looking at the whole second level library of the chemical bonds that are actually on the surface of the 2D materials. And then if you look into transition metals, chalcodenides, you have like even third order of possible chemical reactions that can happen on the surface. So it is really exciting what kind of biosensing you can do with these 2D materials, and the world is really open for that. Okay, I will start with a few examples on optical biosensing, or I would rather call it optoelectronic biosensing, because still at the end we're trying to it's still read out electronically most commonly. So the most conventional optoelectronic or optical biosensing would be to have a MOS2 or some 2D material that you have the biorecognition elements on. So this is MOS2. These are your aptamers or antibodies in this case. And you would shine a light with this light through the prism. You would induce plasmonic resonance on the surface. And then this plasmonic, well, as resonance is very sensitive to anything happening on the surface. So whenever there is a molecule reacts with your recognition elements, your resonance shifts, and you can create a really sensitive biosensor. If we look into a little bit more, let's say, unconventional approach, you could have graphene. In this case, it's a graphene. It's so-called cage sensor. You could have a graphene that you constantly shine light onto. You induce optoelectronic generation of electron-photon pairs in there. And then if you have a heart, so I don't know, it's not visible here, but then they would place a heart here, a whole heart or cardiac cells. And the cells would produce electrical potential that would change the Fermi level of the graphene that is in contact with the heart, and that would in its turn change the optical signal that outcomes from the sensor. So you create a sensor for cardiac activity entirely optical. If you look in a little bit opposite way, maybe not biosensing in this case, it's a little bit more on a stimulation, but you can use graphene and shine UV light on top of it. Well, or in this case at the bottom, if you see. So you shine UV light on graphene, it reacts with the UV light, ultraviolet light, and it generates excess of electrons. And then if there is a cell nearby, I don't know, you might have known about the Nernst potential. So whenever you have an excess of electrons or an excess of charge on one side of an electrogenic cell, you create a depolarization of the cell. So you can actually stimulate the cell to depolarize and to create stimulus to the cell, which is really important for many cellular applications. Now let's go to a more probably the most common 2D material-based biosensor, which is based on field effect transistors. And before we go to graphene or even to liquid-gated biosensor field effect transistor, let's look at the classic field effect transistor. So you would have a substrate, you have a dielectric, in this case it's silicon dioxide, you have a conductive, semiconductive channel, source and drain, and you have a gate. And you apply electrical potential through the gate, which changes the conductance of your channel somehow. On the other hand, and yes, so in those devices will look like literally small pieces of 2D material on silicon. On the other hand, to make really a biosensor, you need this channel to interact well with the liquid most commonly. So you have a liquid, in this case, just a PBS of water. You have the channel, semiconductive channel. You have a substrate, but substrate doesn't play any role here. You apply the electrical potential through the electrolyte. And as in any electrolyte, if you apply potential, you'd form an electrical double layer. And in the case of graphene, there's in 2D materials, you would have an air gap. You'll probably have a quantum capacitance. But you will form an electrical double layer on this side of the channel. And this is where, you know, your analyte can actually react with the channel, and this is where the biosending happens, and this is how you gate the device. And on the bottom right, you can see a physical picture of how those, let's say in this case, it's a graphene, but it could be any 2D material biosensor would look like. Let me skip that. That's so this is a schematic of a biosensor. So you have a source drain, you have electrolyte, and you have a liquid on top of it with functionalized receptors. So a conventional biosensing would be, as you would start with a receptor on top of a graphene surface, and you measure a performance of the device. You have this typical V-shaped IV curve. And then once a charged molecule comes in contact with the receptor, so let's say if it's a positive charged molecule, so an extra positive charge comes to the receptor, so you receive the shift, left shift of the Dirac point, and you have a drop in the current. If an oppositely charged, negatively charged molecule comes to the receptor, you have a right shift of the current, or increase, right shift of the voltage or increase of the current. So that is more of a really conventional biosensing. If we look into a conventional, unconventional approach, you can actually utilize so-called frequency doubling phenomena, which is commonly used in RF applications, but you can do this in liquid. And in case of applying a DC potential to the gate, you can actually apply an AC potential to the graphene transistor with just, let's say, a frequency of 2 Hertz here. But because you're sampling over the Dirac point, you're actually going up and down twice more often. So your output frequency is actually twice higher. And when you have a shift of the Dirac point a little bit, so you're way more sensitive to the shift of the Dirac point in this case than in a conventional biosensing. I will not go too much into describing what kind of analytes have been measured with what kind of 2D materials. This is just a very general overview of those classical biosensing examples. Clearly, people have done a lot of DNA sensing. This is MOS2 2D material, if I'm not mistaken. You can do ion sensing with using ionophores. And actually, in this example, the authors have used black phosphorus, which is known to be not exactly stable, but because there is an ionophore, it increases the stability of the whole device. You can do dopamine sensing, you can do cholesterol, glucose, all kind of sweat or blood composition sensing. And in the last 2 years, we've probably seen a huge rise in all the virus sensing approaches, such as COVID-19, with all kinds of 2D materials, including graphene, MOS2, WSE2. And it is exactly where one of the really niche applications of the 2D materials lie in here. If we look into unconventional solutions, what Professor Akimande recently already mentioned in his talk, the Cardiac Bio have also been selling those, or they producing those commercially chips with the graphene FETs. And what they've recently developed, I believe this work from this year, they actually introduced CRISPR-Cas9 gene, which maybe biomedical community is very much aware of. And they can actually discriminate between different single point mutations in the DNA, almost in a live approach. So this is really very important and unconventional use, almost market-traded technology of the 2D materials as a biosensor. Another very exciting and important part of the 2D material biosensors are cellular, or let's say in this case, in vitro cellular activity sensing. So you can actually grow or bring electrogenic cells, such as, so in this case, you can have neurons, or you can have cardiac cells, because these are the most common electrogenic cells. You can grow those cells on top of the graphene, and then when the cells beat, mechanically beating, or they'll produce electrical signals, there is a change in intracellular potential, as you can see on figure C back here, but there is also change in the extracellular recording. So extracellular potential changes as well that you can pick up with your 2D material sensors. And you can do this. This is a very heavy slide, so bear with me. So you can have this like in vitro, so you can grow cells on top of your devices and just measure like this is a like 1 Hertz frequency beating of the cardiac cells, and cardiac cells form syncytium, so they beat in single direction. So by monitoring, like having an array of the devices, you can monitor where your pacemaker is and where the beating propagation happens. And in the same way, you can stimulate the cells. In this case, you see the cells were stimulated with noradrenaline, and the frequency of beating increases almost twice. And then you can kill the cells by adding SDS. You see there is no reaction. And you can do the same measurement by using neuronal cells. What is even more exciting, perhaps, is that you can actually use a short video. I don't know if there is a sound. There is no noise anyway, so it's just noise. I don't know if you hear it. But these are the microelectrode arrays based on graphene in this case, but it could be any 2D material that are used for in vivo recording, in this case in a rat. So you could implant those devices into the rat to record electrical potentials of the brain. And if I, oh, sorry, that's all right. So you can measure these electrical potentials of the whole brain by implanting those cortical or brain implants right into live animals. And a more conventional approach would be to use microelectrode arrays or graphene field effect transistors, and so 2D material field effect transistors. And there is a lot of work that have done it, and because the 2D materials are transparent, there is really a great use for those, because there of another technique that is called optogenetic stimulation of the cells, and people want to stimulate the areas of the brain right through your measurement electrode, right exactly where you stimulate. Optically, you want to measure electrically. This is where transparency of the graphene and 2D materials play really very important role. Once again, maybe going in a little bit more unconventional approach, what I have here is a so-called switchless multiplexing approach that was developed by the group of Jose Garrido. This is exactly going back to this frequency doubling approach that I mentioned earlier, that instead of driving DC potential by having 2 probes of the transistors, you can actually drive just 2 different frequencies into those graphene devices and just demodulate the signals afterwards. So you will mix the signal and then you'll demodulate them back, and you can record the electrical potential, let's say, from 32 electrodes by using only 16 wires, or let's say 31 wire instead of 64. So this is really a great improvement in terms of scalability of these approaches, because number of wires, power consumption, and these are really important parameters when it comes to implantable in vivo biosensing. Another really interesting and unconventional solution to biosensing and also neuronal sensing in this case, I just like this work. This is really a manifestation of why 2D materials are so exciting for all the biosensing. This was a work, I believe, from already a few years back, where authors have fabricated MXints, titanium aluminum carbide that was selectively etched in HF. And these devices, just bare devices without any additional modification, they just turned out to be sensitive to dopamine. And dopamine is really important molecule that, well, besides the one that makes you happy, it is also important. It's a vehicle that goes between between axon and neuron when the communication happens. So really, and it turns out that this 2D material is sensitive to the dopamine, which was confirmed by the electrical recording and optical recording when the dopamine sensing happens, and when the fluorescent recording of the electrical signal, electrical potential firing is happening. Another very big field of 2D material and sensing is so-called nanopore sensing, that you can create 2D material-based nanopores. So these are very common nanopores, just right now made of silicon nitride. So you have an aperture of 20 nanometers made of silicon nitride. You would transfer MOS2 on top of it, and then you would shape MOS2 so you make a very tiny opening in that MOS2. And then by applying, and I believe in the electroosmotic force through that, and having a DNA in those solutions, you can drive the DNA molecules through these nanopores by recording electrical current, open pore current, excuse me, through the nanopore of 2D materials. And you can record a change in the current that is going through. So, and if you look deeper, there are probably tens, dozens, and hundreds of works on the topic. But people can actually differentiate between different pairs of DNA, different DNAs, and so you can study the DNA quality, DNA composition by driving them through those nanopores. Very exciting. Another very interesting that perhaps I don't call it very much of a sensing here, it's in a quotation you can see, but it's called synaptic sensing. You probably in the last probably 5 years, you've seen a really rise in all the neuromorphic computing, neuromorphic engineering, synaptic neuromorphic devices. These are the devices that would mimic synaptic or would mimic artificial neuronal behavior. And what is really important here is that besides just, let's say, ionic sensing that you can build here, so let's say in this case, this is lithium ion sensor that could, dope, in this case, WSA2, tungsten selenide. You can dope it and you create this neuromorphic behavior, but you can also use it as a sensor, but it's not that exciting to use as a sensor. What is exciting is to use those artificial neurons as a, more like automated decision-making elements, like the neurons, like a mini brains in your automated systems. And one of the very recent works I find very exciting that it goes in this direction. This work published in Science Advances, where they fabricated, they tried to mimic almost the whole natural pathway of optical recordings, neuronal from, let's say, artificial retina to sensory neuron to synapse. They have a firing neuron to have a decision to move a muscle, for example. So they've reproduced the same thing by just constructing different devices on using indium phosphate, input optical signal, and then there is a postsynaptic current, the change in voltage. And then, well, of course, it wasn't really interacted with an actual human hand. It was more like a robotic hand. And if you see that without learning black, so the movement would happen, rapidly. But after this automated training, this automated artificial consciousness, the moving happens almost right after the light is on. I believe we're way far off, to make those devices fully integrated with wearable solutions. But this is one of the most exciting approaches that I think we are going to. And this is exactly why 2D materials are really excellent for wearable biosensing. I am not going to go into this direction here. Professor Kovanda has given a really great overview on graphene for wearable biosensing. We've also done PTSA2 and PTT2 as wearable electronic tattoos, and working on many different approaches, and that is really a very exciting field. I believe this is one of the last approach of where 2D material can be very useful in terms of a biosensing are contact lens. And you can make the contact lens on the left one, on the left graph, you can see it's mainly done with graphene, and it can be used to measure electroretinogram. There are other works, other approaches where people have introduced biosensors in there as well. So let's say a glucose sensor, so you can also measure glucose through your tears, which is correlating to the glucose in your blood. And on the right graph, you can see so almost artificial retinas made of 2D material-based optical sensors. So you can have a whole array of MOS2 as an optical sensor, graphene as an interconnect, and you can shine light and you can make the whole imagery. So you can imagine in maybe years to come, we can have almost substitutes of retina in your eye made of fully 2D materials. And this is really another really great approach where they fit. To conclude, there are two, let's say, I'll go into problems, challenges, and possible solutions in the field. And one of the most common challenge in biosensing, especially liquid-based biosensing, is device screening. Device screening is a phenomena that is depending on the molarity of the salts. So I mean, when you form an electrical double layer, it has a certain height, which depends on the concentration of the, let's say, water or solution that you use. Your normal blood or normal cell line would have an Debye layer of approximately 1 nanometer. So if your biorecognition element below that 1 nanometer, which is rarely, you're safe. Like, I mean, your reaction would work and you'll record it very nicely. But if your recognition element is high, so like you're way above the Debye layer, whatever happens here, whatever charge that is attaching here, you'll most probably not record it. So this is a problem, and that has been there has been a few approaches to that. One would be to create sort of a small shell around these receptors to sort of make the analytes to go into the gel and react with the bioreceptors sort of without undergoing this Debye limitation. Another very exciting, I don't think it has been fully approached yet, but to measure this device at higher frequencies, almost above 10 megahertz. And this is when water, instead of being more like a movable ions, it becomes more of a dielectric. And then you can use it as a normal, you can approach to it as a normal, silicon dioxide dielectric. So you do not have this Debye screening. And the third approach is to actually crumple the 2D material. So you bring these planes of the graphene together, and locally your Debye layer would sort of merge, and it will be much higher than it is supposed to be in this exact electrolyte. And the last, so this is a heavy slide, but I would just recommend it to go to this review work from almost 8 years ago, but I think it is many of those points are still very relevant in terms of application and challenges of the 2D materials, depending on their band gap, depending what 2D material you use, 2D tellurides or insulators or graphene, highly conductive. And then depending on to what approaches you want to use it for, as a dielectric, as an interconnect, as a biological platform. And there are, on the right side, there are common challenges such as cleanness of graphene. What is the mobility of the 2D materials? What is people would want to reduce the CVD growth temperature of the 2D materials, introduce more effective doping. So all of those points on the right are probably still very important research topics that one may think of as to fully study in this regard. And with that, I would like to conclude, and thank you very much for your attention, and I'm open for questions.
Thank you very much for presentation. I have not received any questions. I don't know if anyone would like to ask a question.
I have a question in direct messages. So the question goes, what are the advantages using CMDs for biosensing application instead of using graphene? Yes, that is an excellent question. And the general answer, really short answer, would be a band gap. Because graphene doesn't have a band gap, it's quite a leaky biosensor, so it would be much more power consuming. And then, if maybe looking also 10 years into 20 years in there, we might think of 2D materials fully substituting a whole CMOS, or maybe 30 years, fully substituting the CMOS technology. So you could think of a full 2D material based approach as a biosensor and logic and amplifier signal. So making the CMD based sensing elements as well would be very important in that regard.
Perfect. Thank you so much. Does anyone else have? You have more questions in the chat?
I have one question. So given that these biosensors rely on a 2D material with contacts, compared to applications in electronics where the contact resistance is one of the major challenges right now, how much is the contact resistance affecting these biosensings? Is the signal to noise usually large enough that's actually not that big of a deal, or is it something that is an impediment?
Yes, well, I would say the signal to noise ratio is high enough here that it doesn't affect the recording too much. And again, we're also working here mainly with rather large devices and like more CVD grown devices. So, I mean, maybe if you would like scale them down to like a few hundreds of nanometer flakes, then the contact resistance could potentially be problematic and they had to be considered. But currently, that's not really a big problem for biosensors in the regard.